It's my pleasure to introduce Professor Tiina Roose from University of Southampton in the UK. Tiina has a background in mathematics and modeling and systems analysis, and I know during one of her postdocs, which is at Harvard Medical School, I think she said she was working on fluid flow through the human vascular system, but we're very fortunate that she came over to agriculture and got interested in soils and roots, and is a great collaborator also. So Tiina's going to talk about multiscale image-based modeling of plants and soil.
So alternatives need to be found. And we do know that you can extract phosphorus from human wastewater treatment plants and things, but those fertilizers will be more expensive. So no matter what scenario we are in, we should be optimizing how we deliver and grow our crops with a more economic way of delivering phosphorus. And there are two different ways we can do. This is by optimizing plant traits, by breeding, for example, or optimizing soil amendments. So what we've been doing in Southampton, our approach has been to develop these mathematical models to run lots of trials in silico. But the challenge, of course, is that we need to make sure that these models are scientifically accurate enough to make these predictions. So because when we first started doing our phosphorus models, the biggest bit that was the most influential parameter was always the root structure and how fast it grows in soil. So very quickly we went into the land of imaging, because that's the one method that allows us to see what's happening in the soil, because it's opaque. So I don't need to tell you what 3 or 4D computer tomography is. So we have a big facility in Southampton which has multiple different tabletop machines. So you image, you get their images, you segment them out. So you get the structure. Now that just is sort of, for engineering speak, a pretty picture. It doesn't actually say to you anything about the function of that system very much. So we take those images and assign function to how, for example, solute movement or water movement occurs in the structure that we observe. And then use this information from functional modeling to then plan the next experiment. The other way of looking at that image-based stack is sort of inspired now from biomedical sciences. Again, you're imaging, you're segmenting, but then you take two-pronged approach to modeling, in a sense that one is this image-based meshing and doing a very detailed mathematical modeling. Now, if you're in practical situation, you can't do it because that's very labor-intensive, very computation-intensive. What medics do is they extract fast morphometric measures out of it, and then use that measure. Now, with plants and soil in particular, for plant function, we don't know which fast morphometric measure is the correct one for particular function. So by doing the two-pronged approach, allows us to find the fast approach and slow and the very scientifically rigorous approach to bringing them together. But what is the big problem for soil modeling is this, that the soil is multiscale. And we've already seen various different examples of this. This picture is from a recent review paper we did with EMSL, so you have to basically, there are different phenomena happening from a molecular scale, from very fine molecular scale to cellular scale to field ecosystem and globe scale. And the way the modeling now fits into this thing is that modeling frameworks are there to integrate all the knowledge from these different experimental techniques. And virtually on every scale, the X-ray techniques are involved, aren't they? We've already seen some of these synthetic habitat work described here yesterday. So modeling will bring together all the different techniques. Now, the mathematicians on that right column there have been doing this multiscale modeling and talking and doing it for a good 30, 40 years, I would like to say, but quite in isolation from experiments. What became clear in 10 years ago, that this multiscale analysis that we and I'm a mathematician, so I'm guilty of that as well, is that we float up and down those spatial scales with our mathematical equations, but that floating up and down, translating information is only as good as it if you have to start calibrating those models at each of the scales as well. And so here's the other way of looking at these different techniques. And those are the techniques we use in lab in Southampton now. And we noticed that on a finer scale, we start with X-ray-based techniques. And then as we go bigger, our techniques have to change by nature. We've tried neutron radiography and tomography, and then we also build soil sampling, sort of microfluidic devices for soil sampling. And so I will give you a couple of very quick examples of what we have done. One is now one of the kind of first studies we did, which we did it really for fun by contrabanding to another synchrotron session on materials, aerospace materials area. So we wanted to know root hairs that are known to be hypothesized as being very important for phosphorus uptake, especially of strongly bound nutrients as phosphorus. So they penetrate the soil pores, but we wanted to know, is it better to have longer hairs or shorter hairs, more denser hairs? What's better? And we developed lots of mathematical models, and we realized that it all hinged on knowing the morphology of the hairs with respect to soil particles very accurately, because most of the hair morphology measurements were done in petri dishes, like you see here. And it's intuitively obvious that if you put soil particles in front of it, the morphology would be different. So we, as I said, contrabanded the samples with this little assay, which is 3D printed syringe barrel assays. Root hairs are roughly 10 microns across. They can be up to 1 millimeter long. We needed synchrotron because whilst the tabletop machine can reach those 1 micron resolutions, it comes at the expense of time. And plant roots, whilst you kind of intuitively think they're not very active, they actually grow 2 centimeters a day. So if you're dealing with a 8-hour scan, in that time the plant root is kind of actually moved quite a bit. So we went to synchrotron and imaged and got these sort of images on how root hairs interact with the soil particles. We took those, we put the standard first-order kinetic phosphorus model onto it, where we have the phosphorus binding to the soil particles, phosphorus diffusion in a pore space, and then uptake by root and root hair surfaces. Now, what this allowed us to then, running that simulation, allows us to say now, like how local is, for example, this phosphorus uptake. It's always hypothesized because they go into smaller pores that it's local, but we can now say on the right here that it's roughly half of the area compared to where the root itself is taking up phosphorus. And it also says that what the function of root hairs is not to dominate phosphorus uptake, but they are roughly doubling phosphorus uptake, at least for this imaging system that we had. The root that we imaged, that's the case. So this encouraged us to start looking at the other fine filamentous things in here. So inspired by our success at imaging root hairs, we went straight into mycorrhizae. In hindsight, maybe we should have done saprophytic fungi that aren't quite so dependent on a plant, but we went and did it. So mycorrhizae are obligate symbionts, so they have to grow with a plant, which poses a challenge of how do you image them, especially using synchrotrons. Now, we in engineering department, so engineering students really like designing different devices. And so this is the sort of mycorrhiza imaging assay growth box that we designed. It's designed to fit into aeroplane overhead compartments so that we can drive around with it. It has, as you see here, these little water tanks where we can, with a wicks, keep the syringes, which is where the fungal compartment is, well hydrated. The problem with these very small samples is that they can dry out very quickly. So using the wick in the syringe technique, we can keep them under constant water supply. So plant is in these compartments, as you see here. There's a mesh between, so plant roots can't grow into side branch, these little syringes here, but the fungus can. So the fungal inoculation was done in a root compartment, and then we observe what's in these little side compartments. Again, this is now our examples that we obtained. There's a lot of still open. I mean, that study created more questions than it answered. The big questions are, what are these little blobby things on the top that you see? Are they water droplets? Are they amoebae? Are they some exudates? What are they? We still don't quite know. If we look at the X-ray images of these in control, where we know there is no mycorrhizal fungus, we see strikingly, I hope you see that there's a sort of structure of fungi that we see in the soil is quite different between inoculated samples and uninoculated control samples. Control samples have lots more spore, like these little blobby things, whereas Rhizophagus inoculated samples have a lot more regular sticks and straights. So our study combined basically two treatments, mycorrhizal inoculum, no mycorrhizal inoculum, added P in a fungal compartment and no added P. And what we did was we X-ray CT scanned in 3D those samples, and then we took those same samples, resin fixed them, and took a cut across to do the XRF elemental mapping of it for phosphorus, sulfur, and then we can correlate that 2D map back into 3D in X-ray computer tomography, so that and get the distance maps that are genuinely 3D distance maps from the fungal hyphae. Now that correlation sounds simple, that, well, you're finding a flat thing in a 3D stack, but it's not quite as straightforward. It's quite computationally expensive, but it allows us to now quantify the amount of phosphorus sulfur as a distance from mycorrhizal hyphae that we see in 3D. And also, obviously, as a distance from root compartment. Now, long story short, we basically, with this study, even though it opened more questions than it answered, we could narrow down the mycorrhiza hyphal P uptake parameters. We did correct them by roughly an order of magnitude, and that was because old models, we were sort of guessing what that parameter might be. That was consistent on a field scale studies. Now, that is progress as far as I'm concerned. So conclusions from this bit is that the root hairs, we can determine root hair and mycorrhiza P uptake parameters with a lot more accuracy. Root hair P uptake is on the same scale as diffusion in the soil for P. We did the same for hyphae, and also as a part, because we imaged the sulfur, we think the sulfur is a surrogate marker for mycorrhizal hyphae now in the soil phases where we can't structurally separate the hyphae out. So we're looking into more of that sulfur story. But moving on to a bigger scale things here, we are now looking into really into developing mathematical models for the mechanics of soil, because we all know that as the climate is changing, soil is getting harder possibly when it dries out, so it becomes more rigid. It might be that the crops have harder time growing into the soil mechanistically. So imagine that these, where you have a little red circle, is when the root goes near, you can actually sort of be able to visualize how it empties the pocket of water as the root grows nearer and how the particles move. Now in here, movie is working, this is the same image now, where we do the digital volume correlation things to see how things are moving. And what we see is, let's go again, is that the root is growing like a little corkscrew into it. It isn't pushing itself in, it is corkscrewing it in. So the arrows are the strain arrows around it. Now, why we're looking at it in particular is that in the UK, we want to start building the models of soil slope failure. So National Rail in the UK had for years and years a problem of leaves on the line, which why all the trains were always late in autumn, because autumn arrived unexpectedly. So to deal with that problem, they cut down all the trees on the railway lines and slopes. But now, because the roots are not there in the soil to maintain hydraulic tension in the soil, we have a problem of landslides. And it's becoming a lot worse. So what we are doing is we're doing centrifuge experiments with the planted soils, but we're also doing shear box experiments in the X-ray CT scanners to separate out what the different what these different mechanistic aspects are. That what's the aspect of roots reinforcing mechanically the soil versus the roots keeping the soil particles under tension in terms of water extraction. And what we are finding is that the main factor actually is the tension bit, not the fact that they are there as a sort of fiber reinforcement things. It's actually the fact that the plant is transpiring in the soil that is the main stabilizing factor. I want to move on now because I think I'm running out of time. And I want to talk, Dean kind of showed that a little bit, is the Xylella disease we started looking at. So Xylella disease is a major, major problem that Europe has now woken up in the last 10 years about. Xylella is bacteria that is carried by mosquitoes from tree to tree. And it has 500 bacteria species that are susceptible to it. So all the kind of fun things like grape wines and plums and oranges, coffee and almonds. It arrived in Europe in 2013 with a souvenir coffee plant from Costa Rica by a tourist who just took it with them. And now it's killing these olive orchards in Italy. And yet, what you see here is on the left, there is a regular olive orchard, which is all dying off effectively. And on the right, there's a susceptible variety of olive trees. And nobody quite knows what things sort of contribute to this susceptibility versus resistance. So the way the disease progresses is it's a bacterial biofilm that clogs up the xylem vessels. And you notice it first, the disease as a drought symptom, which is very difficult if you're in southern Italy to separate from actual drought system. And so we looked at it in olive and citrus trees. Resistant and susceptible varieties, and because in South America it's out in a lab, we very kindly got time on the Mognobeam line in Brazil, where we could scan those diseased and resistant plants, which we're not allowed to do in Europe because it's under control. So So our sequence basically was similar to what we do with soils, that we image, we get the raw images, we then get the segmented information from image processing pipeline. Then we do the subsequent sort of macro scale fast analytics on that data. But then we take the images and we develop a mathematical model based on those images, where we have now how does the bacterial biofilm sort of evolve in a xylem vessel. So the red blobs there are bacterial biofilms, and blue is just the water. And ultimately, we basically get this circle of modeling that is modeling combined with X-ray geometry. It's a sort of digital twin of Xylella disease in the olives and citrus trees. And to cut a long story short, what we found that between these 4 different types of samples that we were at high resolution able to image in Sirius, in case of citrus diseases plants, we didn't see any huge statistical significant differences in xylem architecture between the 4 resistant, susceptible, infected, and uninfected ones. But the resistant ones did tend to have smaller. It's just it wasn't significant. In olives, we did see a statistical significance between the resistant varieties and susceptible. And also healthy versus infected ones had a bigger lumen. So what this suggests is that the disease progression is different in citrus trees and olive trees, that in olives we can actually possibly breed for resistance by manipulating the xylem architecture. In citruses, the story is a little bit different and requires some other mechanism that provides resistance there. And that is so. Let me finish now here. So image-based modeling is quite labor-intensive thing to do, because you need to be able to do the image segmentation, and then also integrate it with mathematical modeling to get your digital twin. But these are the alpha models. These allow you to test the most detailed experiment, sort of scientific hypotheses on this. And they also will allow you to validate the larger scale, faster models. Like we all know there are crop models out there, but they have lots of fuzzy assumptions in them. But we can use these image-based models now to build confidence in these large scale, sort of a lot simpler, faster models. Crop models. I finish here. This is my main collaborators throughout the years, and this is my group of postdocs and PhD students. Nancy, who is here third from the right, she came into the lab as pure mathematician, never done an experiment or anything, and then she started doing scanning and experiments, and now she's very passionate about the X-rays, CT scanning, image analysis, and has her dream. She's hopefully goes to work in Sirius. And these are the main funders and the facilities we've been using.
Thank you so much, Tiina. That was excellent, as always.